Welcome back. So in the last video, we talked about the function of the ribosome. And we're actually going to do a little bit of application to the ribosome here. And that's really the function of the rough ER and the Golgi apparatus. So let me get my pen here. Okay, so in this video, what we're essentially going to do is talk about two organelles that exist inside the cells of mammals, and all eukaryotes for that matter. And this is going to be the function of the rough endoplasmic reticulum, which people usually abbreviate as rough ER or RER. I'll usually abbreviate it like this. And then we're also going to talk about the function of the Golgi apparatus, or sometimes they'll call it the Golgi body. And a little bit is known about the Golgi apparatus, but not a lot. The Golgi apparatus is something that's kind of hard to study, and we're not going to go into excruciating detail on it because, quite frankly, if you look in your anatomy and physiology textbook or your biochemistry textbook, depending on what context you're watching this video in, there's not a lot known about the Golgi apparatus, but there is quite a bit known about the rough endoplasmic reticulum, so that's going to be where the majority of this video is dedicated, honestly. Okay, so this picture right here, this is a pretty typical picture of a cell that you can find on Google. And I want to first note some things about this picture. Number one, if you look at the various organelles, you can kind of see, hopefully, that in many cases, in pretty much all the cases, they've sort of sliced off the top of it. In the case of the nucleus, they cut off 1/4 of it. But in most cases, they've sort of cut off the top half, so you can kind of see the inside of the cell. So if you look at the mitochondria, hopefully you can see that they've essentially cut off the top of it, so you can see inside of it. If you look at the rough endoplasmic reticulum, hopefully you can see they cut off the top of it. They did the same thing with the Golgi apparatus. Now, here's something interesting that hopefully will make a little bit of sense to you. They've actually drawn this quite accurately. One thing about the rough ER is the rough ER is actually continuous with the nuclear membrane. So the nuclear membrane is just basically the outer surface of the nucleus. And the rough endoplasmic reticulum kind of branches off from that, and you have the rough ER. Now, when you're looking at the rough ER, I'll try to highlight it in here a little bit. If you sort of look in here, what I'm essentially highlighting in here, this is essentially what's known as the lumen of the rough ER. Okay, so if you come over here to this picture, they've sort of drawn this kind of really generally, really roughly. And this inside of the rough ER, this is called the lumen. So the lumen is essentially what I highlighted in yellow. It's sort of the inside of the organelle. And inside the organelle, there are enzymes. There are enzymes inside the lumen of the rough ER. And we'll talk about the implications of that later on in this video. But what we're essentially going to do is we're going to talk about the function of the rough ER. And in general, what the rough ER is it's a membranous organelle that lies just outside the nucleus, and it's responsible in part for some of protein synthesis. Okay, the actual enzymes that synthesize proteins, those enzymes are the ribosomes. That's what we talked about in the last video, ribosomes. Those are actually the enzymatic machinery that make the proteins. But it turns out that some of these ribosomes can actually attach themselves to the endoplasmic reticulum. And in fact, if you were looking at a microscopic picture of the endoplasmic reticulum, you would see some of it is really smooth and others appear to have these bumps on it. And the little bumps that you can essentially see kind of right here, hopefully you can see the bumps on there, those bumps are actually ribosomes. Okay, and some of the ribosomes will actually attach themselves to the ER membrane, and this is where protein synthesis will occur in some cases, and we'll talk about what kinds of proteins are made from there. Okay, so without boring you any further, let's actually look at this picture. It's kind of imposing, but one thing I've sort of found over the years is there are, visual learners, auditory learners, and so forth, and I try to appeal to all of that. Sometimes when you're talking about the rough ER and the Golgi apparatus, sometimes I don't think teachers really elucidate to you the functions as they occur in real time. So hopefully we'll get kind of an intuitive sense of this. So in the last video we talked about the ribosome, and the function of the ribosome actually still applies here. So let's actually look at it. Okay, so just kind of to orient you with the picture, this green stuff that's right here, this kind of green stuff, that's the mRNA. And notice we have the 5' cap right here. If you remember when we talked about mRNA processing, you have polyadenylate polymerase that kind of adds this adenylate, this AAAAA and so forth sequence to the 3' end. And then all of this green business in here, that's just the mRNA. Okay? And we have the ribosome that attaches itself to the mRNA and starts translation. So this process right here that they're talking about, this is the process of translation. Translation is using mRNA, tRNA, and ribosomal RNA to synthesize a protein, and the actual process is carried out by the ribosome. Now, one thing that's really critical to understand is part of the mRNA codes for something called a signal sequence. And we're going to go in some detail about the signal sequence. This kind of thing that's in this bluish purplish color, this is what's referred to as the signal sequence. And the signal sequence has a really important function in the synthesis of many proteins. We'll talk about that in a minute. But then you also see this whitish part of the protein coming off. That's not the signal sequence. That's the remainder of the protein. So basically, in the case of proteins that end up inside the rough ER, the signal sequence is the first part that's synthesized, and then everything after the signal sequence, that's the remainder of the protein. Okay, so without going any further, in terms of the rough ER function, I want to show you this picture. This picture is not something you're responsible for if you're in A&P. This is something that maybe will give you an idea of what the signal sequence is. But if you're in the context of biochemistry, this is definitely something you have to know. Okay, so the signal sequence is highly variable depending on which protein you're talking about. But there does seem to be really a commonality in all of this. Basically what you have in general is, and let me kind of bracket off the components that I'm talking about. So starting here and going there, starting here and going to here, and you could keep going on and on like this. But what it really seems to be is there's approximately a sequence of about 16 to 20 hydrophobic amino acid residues. So in general, when you look at these sequences, you'll notice here's a leucine, an alanine, leucine, alanine, leucine, tryptophan, certainly hydrophobic. You have all these hydrophobic residues, and on average, it's about 60 to 20 of them. Okay? And this exists basically on the N-terminus of the protein. Okay? And you have this long sequence of hydrophobic residues. Okay, so these hydrophobic residues over here on the left side of the cleavage site, all of these are called signal sequences. And if you look at the sequences, there doesn't really seem to be very much sequence homology. But suffice to say, what happens is when the DNA gets transcribed, of course you get mRNA, right? And the mRNA consists of exons, right? It consists of exons and introns. The introns, of course, don't code for protein, but the exons do. Part of the function of the exons when they get translated is to code for these signal sequences, and we'll talk about the functions of those later. I won't mention it right now, but this cleavage site, this is the site of cleavage for an enzyme called signal peptidase. We'll talk about that in a few minutes. This is the cleavage site for signal peptidase, a very important enzyme in rough ER function. Okay, so I've mentioned that we have these ribosomes that attach themselves to the rough ER membrane. Let's talk about what happens. Okay, so I have this ribosome that currently is really sort of floating in the cytosol, and it's making a protein that has a signal sequence. And after the signal sequence, there's some other protein that's synthesized after it, still attached. Well, what's essentially going to happen is, in this case, there's going to be another protein. Okay, there's another protein called a signal recognition particle. This SRP is called a signal recognition particle. Signal recognition particle. And the signal recognition particle, notice, will attach itself to the ribosome. Okay, so there's sort of a binding site on the ribosome for the signal recognition particle, or SRP, to attach itself. Okay, and the signal recognition particle is going to play a role in attachment of the ribosome to the rough ER. Okay, now here's some important things to understand. Okay, I'm going to use hopefully a good analogy here that'll help you understand kind of how this works. So before we get really any further, I have to sort of define what a receptor is, because we're going to run into some receptors. So we have this big concept in any kind of biology course of a receptor. And hopefully this analogy will kind of make sense. And I know I have some viewers who are not from the United States, so I kind of have to explain this a little bit. So we have this sport in the United States called American football. Okay, and the idea behind American football in very general terms is you have this guy called a quarterback. And the quarterback in many cases throws the football. Okay, so the American quarterback throws the football, and you know the football's spiraling through the air, and you have this other position for a guy on the same team, and he's called the receiver. Okay, so you have the quarterback, abbreviated QB, and he sort of throws the football. Okay, and over here you have the receiver, right? And the job of the receiver, his job is to catch the football. Okay, his job is to catch the football. And he's done his job if he catches the football. So the receiver is sort of like the receptor. His job is literally to catch and grab a hold of something. Okay, so when we're talking about the analogy to molecular biology, physiology, and all that stuff, the receptor's job literally is to bind something. Okay, well, what does it bind? Well, the receptor could really bind anything as long as it's a molecule. So the receptor, what it literally does is it binds a molecule. Okay, and typically the molecule to which the receptor binds, we typically call that a ligand or a ligand, however you want to pronounce it. So the ligand is just the molecule to which the receptor binds. Okay, so for example, if I have a ribosome receptor, the molecule to which the ribosome receptor binds is the ribosome. If you were looking at it kind of with the analogy to football, the receiver would be the ribosome receptor. The football would be the ribosome. If I'm looking at it with respect to the SRP receptor, the receiver would be analogous to the SRP receptor, and the football would be analogous to the SRP molecule. Okay, so hopefully now that we kind of understand what a receptor is, it's just a molecule. It's just a protein usually that binds a specific molecule. Okay, so hopefully that makes sense. And what we essentially have here is we have two receptors that are in the rough ER membrane. And I'll circle these in green. So this first receptor that's in the rough ER membrane, this is called the SRP receptor. And hopefully, based on that name, you could sort of guess which molecule it binds. It binds the SRP, which is now attached to the ribosome. Okay? And then we also have a ribosome receptor in the rough ER membrane, and the ribosome receptor binds the ribosome. So hopefully that makes sense. So what we have basically is we have this ribosome receptor and this SRP receptor in the rough ER membrane. And literally what they do is when the ribosome sort of floats by, they grab onto their respective ligands. So the ribosome receptor binds the ribosome, and the SRP receptor binds the SRP molecule, the signal recognition particle. And so they hold on to it very, very tightly. Okay? And during this time, the ribosome is still translating the mRNA into protein. And now I'm going to use another analogy in just a minute. Okay? What I want to mention at this point is that this SRP, the signal recognition particle, this is basically a G protein. Okay, this molecule is a G protein, so it has to bind GTP. Okay, that's what this step is showing. So the signal recognition particle binds GTP. That's not really that important. Okay, but just understand that in this case, the signal recognition particle has bound GTP, which is just a nucleotide. Okay, now here's the important thing. Okay, I want you to imagine several things. Okay, we have this other protein that's also in the rough ER lumen. Okay, the other proteins were more on the cytosolic side. This enzyme, which is shown here, this peptide translocation complex, this is really on the lumen side. This is on the luminal side of the rough ER membrane. And here's the idea. Okay, let's use the analogy of you have a tear in your shirt, tear in your cloth, and you have to sew it back together. Well, let's think of the things that you're going to need. Well, you're obviously going to need your hand, right? So in your hand, usually what people do is they put the needle when they sew it between their thumb and their index finger. I think people have an intuitive sense of that, okay? And you also need a needle, okay? And what does the needle do? Well, it does two things. The needle is attached to usually a long string. And the other thing that the needle does is it sort of guides the string into the cloth, right? You stick the needle into the cloth and pull it through, okay? We're going to have analogous things in the rough ER function, okay? What I want you to do is imagine the peptide translocation complex as your thumb and index finger, okay? Imagine the peptide translocation complex as your thumb and index finger. Imagine the signal sequence, okay? The signal sequence is that thing that's kind of it's in purple and blue, this thing right here. If you were looking at it from the biochemistry context, it's all of this stuff over here. These are the signal sequences, okay? All that business, okay? And remember, they're variable for each type of protein, okay? But you have the signal sequence, that's the needle, okay? That's the needle to which the string is attached. And then the rest of this protein, which I circled in red, so I'll do it again. The rest of this protein, that's the string. So the rest of the protein, that's the string, okay? The signal sequence, all this, the signal sequence is, oops, let me undo that. Undo. The signal sequence, that's the needle. The signal sequence is the needle, and the peptide translocation complex, that's your thumb and index finger. Okay? So what happens? Well, the thumb and index finger always grab the needle, right? So literally, analogously, the peptide translocation complex binds the signal sequence, just like your thumb and index finger grab the needle, okay? Now, what do your thumb and index finger do? Well, they sort of pull the needle through the cloth. Well, you can think of the ER membrane as the cloth, right? So literally what the peptide translocation complex does is it grabs the signal sequence and starts pulling the protein into the rough ER lumen. Notice what we've done here. We basically took a protein that was outside in the cytosol, and what did we do? Well, we essentially, let's do it in light blue. We essentially pulled this protein inside the lumen of the rough ER. And what allowed us to do that? Well, the peptide translocation complex pulled on the signal sequence and literally pulled it into the rough ER lumen. Okay? Now imagine, the needle that you have is attached to a long string, right? So the string is essentially the rest of this protein, right? All of this business over here, this white stuff, that's the rest of the protein. That's your string, right? That's what's essentially going to be the final product. It's going to sew your hole in your cloth or your shirt together, right? But now I want you to imagine something else. Imagine this, okay? Whenever you sew, does the needle get incorporated into your final product, which is your shirt? And the answer is obviously no. If you have the needle that's still part of that, you put the shirt on and you get cut and all that stuff. You start bleeding and it hurts, right? So logically, you essentially cut off the needle. You usually use like a pair of scissors or something and you cut off the needle. So that's essentially what's going to happen here, okay? There's an enzyme referred to as signal peptidase. Okay? There's an enzyme referred to as signal peptidase. Okay? So essentially what's going to happen whenever you get the protein made, this enzyme, which is shown in light blue, this enzyme right here, this is called signal peptidase. What it's essentially going to do is cleave off, notice, it cleaved off the signal sequence. So now this needle is no longer a part of the final product, all that protein. So all this protein right here notice it's getting longer and longer, right? Because we're making more and more protein. This final string is no longer attached to the needle. We hydrolyzed off, or we cleaved off the signal sequence. So what that essentially means is that all the signal sequence was for, these sequences of amino acids down here, these literally, all they were for is literally just guiding the protein into the rough ER lumen. That's all they were for. Okay, so literally, the rough ER uses the ribosome receptor and the SRP receptor to bind the ribosome and SRP molecule, and then the peptide translocation complex binds the signal sequence and guides the whole protein into the rough ER lumen. Then what happens essentially is, once you've got it bound, once the ribosome is tightly bound, essentially what will happen is the SRP, it's a G protein, remember, it has GTP now. Essentially what happens from there is it hydrolyzes the GTP and dissociates. So notice, once you hydrolyze GTP into GDP and phosphate, notice that the G protein, the SRP, dissociates. Okay? And it's free to pick up another GTP. But that's beside the point. But notice that the SRP receptor and ribosome receptor facilitated the binding of the ribosome to the rough ER membrane. And once you do that, the peptide translocation complex grabs a hold of the signal peptide or the signal sequence and essentially facilitates the pulling of the protein as it's being made into the rough ER lumen. Okay? And then, what's going to happen from there is the protein gets longer and longer because the ribosome is moving along the mRNA, facilitating translation. And basically, as soon as the protein is made, the signal peptidase cleaves off the signal sequence, and now you have your final product. You have your protein minus the signal sequence in the rough ER lumen. And then the ribosome, notice, the ribosome dissociates. Okay. Once you use the signal peptidase and the protein's finished, you hit your stop codon, right? You hit the stop codon, and then you get release factor. We talked about that in a previous video, and the ribosome dissociates. Okay, so all the rules of the ribosome are still applicable here. It's just the difference is this particular type of ribosome in this context binds to the rough ER membrane. So literally, this picture right here, of course it's a cartoon, but you have these ribosomes that bind to the rough ER. And I think a lot of times they leave out the important details that kind of help you give you an understanding of this. Basically, the ribosomes do this process and they bind to the rough ER. And then just like your thumb and finger, they facilitate the movement of the needle into the cloth, along with the string, the peptide translocation complex binds the signal sequence and facilitates the movement of the signal sequence plus the rest of the protein into the rough ER lumen. And as soon as you hit that stop codon, right, the ribosome dissociates and is free to do another translation of something else, okay? And the signal peptidase cleaves off the signal peptide, and now you have your final product, which is the protein that is now free of the signal peptide. That is the process by which the rough ER facilitates protein synthesis through the act of the ribosome. Okay? Now, for the purpose of the exam, I really don't care if you know about the GTP and the GTP hydrolysis, but I do want you to kind of understand that the SRP, the signal recognition particle, binds to the ribosome, and then that facilitates ribosome binding to the rough ER membrane through the ribosome receptor and the signal recognition particle receptor, or SRP receptor. So hopefully that makes sense. Now, what we essentially just did is we looked at the signal sequences, we looked at all the things that happen with the ribosome binding to the rough ER, protein synthesis. All those rules still apply, but what I want to do now is look to see what happens to the protein inside the rough ER lumen. Okay, so it's not just as simple as the protein's made and then we're done. What I want to instill in you is that there are enzymes. Let me do it like this. Let's say we have enzyme 1 here, We have enzyme 2 here. Maybe here's a third enzyme, right? There are various enzymes in the rough ER that you don't need to know about, okay? But you do need to know there are enzymes in here that process the protein. So perhaps maybe we add a carbohydrate residue here. Maybe we add a carbohydrate. Maybe over here we add another group. Maybe we add some kind of lipid. Okay, the point is that there are various enzymes in the rough ER lumen that process the enzyme and make various transformations. Okay, so initially in this class I was planning on doing collagen synthesis, but I'm actually going to change what we do in week 3 and do mitosis instead. But in collagen synthesis there are enzymes that process things like pre-pro collagen. They process the precursor to collagen, and there are enzymes like lysyl hydroxylase, prolyl hydroxylase. There are some other enzymes as well, like glycosyl transferases. But the main idea that I just want you to understand is there are enzymes in the rough ER lumen that process the protein further, okay, and that modifies the protein and gets it more activated, okay. Now, what happens to the protein from there once it's been modified in the rough ER lumen? Well, from there, it's going to go to the Golgi apparatus, okay? So sometimes I think when this stuff is taught, they kind of treat all the organelles as kind of being separated from each other. But really, the Golgi apparatus is very closely connected to the rough ER. So let's actually look to see what happens, okay? This right here, this is a picture that you need to know, and it's definitely going to be in your PowerPoint, so look for it. So this stuff over here, all this business right here, all this business, that's the rough ER. You can see the ribosomes attached to it, hopefully. Okay? So what's literally going to happen is the rough ER is going to package the protein into a vesicle. Okay, so notice, I'll try to circle it right here. The protein is basically inside a vesicle. The vesicle buds off from the rough ER. So here's another vesicle budding off from the rough ER. So basically, this protein right here, let me go back. This protein that's here that I circled in red, that I starred, that protein will eventually be packaged into a vesicle, and it'll sort of come off from the rough ER. And notice that vesicle is essentially going to dump, the protein into the Golgi complex, or the Golgi apparatus. So this structure right here, I typically do Golgi complexes in green. That structure right there is called the Golgi apparatus. And so literally, the protein is inside a vesicle and so forth, and it's packaged and moved from the rough ER ultimately to the Golgi apparatus where it's dumped into. Okay? Now, just like in the case of the rough ER, the Golgi apparatus also has enzymes in it. So maybe this is enzyme 4, maybe enzyme 5, enzyme 6, enzyme 7, all that. There's enzymes inside the Golgi apparatus that further process the protein if need be. And once that occurs, then you have essentially the protein packaged once again in a vesicle. Okay, so here's another vesicle that has the modified protein. Here's another vesicle that's budding off that has the modified protein. So the idea is that you have the enzyme inside these vesicles, and it will go to some location, okay? So here is a critical point to understand, okay? Usually when you have a ribosome, okay, when the ribosome is free in the cytosol, okay, so in other words, if I had ribosomes, say, out here somewhere, so ribosomes out here, in other words, ribosomes that aren't bound to the rough ER, they still make proteins, but generally those proteins that are made in the cytosolic ribosomes, they're destined to be in the nucleus or free in the cytosol, okay? But when you have proteins that are made by ribosomes on the rough ER, they have a different destiny, okay? Those proteins tend to go to membranous organelles or the plasma membrane. So I'm just showing you basically two examples, okay? But essentially, if I have a protein that was packaged by the Golgi apparatus and goes into a vesicle, it's very possible that that protein could end up inside a lysosome, for example. Or the protein could have a different fate. So out here, this is the plasma membrane. For example, that protein that's in the vesicle that came from the Golgi, it could also go into the plasma membrane. And there's various other organelles like peroxisomes and mitochondria and so forth. But the end result is that if basically you can sort of think that if the protein got made by the rough ER ribosomes, it's going to go into a vesicle and travel to the Golgi apparatus where it gets more enzymatic modifications. And then it gets packaged into another vesicle, and it usually either goes to the plasma membrane or the lysosome or the peroxisome or the mitochondria and so forth. So basically it's going to a membranous organelle. It could also go to the smooth ER. The smooth ER is another example, or the rough ER if it's one of those modification enzymes of the rough ER. But the whole point here is that if you have a protein that's processed by the rough ER and the Golgi apparatus, generally it goes to a membranous organelle. It's not free in the cytosol or the nucleus. Okay? So hopefully that makes a little bit of sense. And if you're in the context of biochemistry, these right here, these are sequences that sort of guide the protein into different areas of bacteria. So I want to specify that these are bacterial enzymes and proteins. Bacterial enzymes and proteins. But sort of the same kind of rules apply. Okay, so for example, this sequence right here, okay, you have a protein called phage FD. This is phage FD. This protein is guided into the inner membrane through this sequence. So this sequence that I bracketed in red, that sort of guides phage FD, major coat protein, into the inner membrane. Okay? Another example, a very important thing, this is beta-lactamase, a really important enzyme that you would get to sort of in the context of an immune system class. So this sequence right here, from methionine over to alanine here, this sequence guides this particular protein into the periplasm. Okay, so you have different sequences that guide the protein into different regions of the cell. This is for bacteria, but the same rules are going to apply for the membranous organelles of eukaryotic cells like mammals. Okay, so you don't need to know this for A and P, but this picture right here, this is a really good picture that kind of sums up the function of the rough ER, the vesicles that go to the Golgi apparatus, and that eventually go to membranous organelles like lysosomes, peroxisomes, the plasma membrane, and so forth. Okay, so hopefully that makes a little bit of sense, and hopefully it makes a little bit of sense of how the rough ER works. So let's do a very quick recap. Just make sure you understand that some ribosomes will attach themselves to the rough ER. Let's do a recap. So what's going to happen essentially is the same rules are going to apply that we looked at in the last video. The ribosome is going to bind to the mRNA. It's going to start translating, and the first thing it starts translating is this signal sequence. Okay, and remember those signal sequences in the context of biochemistry are shown right here. Okay. So it starts making the signal sequence, and this thing that I circled in red, okay, this is not the signal sequence, but it's still part of the protein. Then you have this ribosome is going to pick up a signal recognition particle, and the reason it does that is because the signal recognition particle facilitates binding to the rough ER membrane. So in the rough ER membrane, on the cytosolic side, you have an SRP receptor. You have an SRP receptor that binds the signal recognition particle, and you have a ribosome receptor that binds the ribosome. So you have dual binding to the rough ER membrane. And then I did the analogy of the needle, and you're running the thread through the cloth to sew the hole in your clothes, right? The peptide translocation complex is the thumb and index finger, right? The signal sequence is the needle, and the string is just a remainder of the protein that's being synthesized. And the peptide translocation complex grabs a hold of the signal sequence and guides the protein into the rough ER lumen while the ribosome continues to translate. Okay? And then what's going to happen is eventually you'll get the whole protein into the rough ER lumen. You'll hit a stop codon, right? That'll code for a release factor, and then the ribosome dissociates. And then we mentioned that when you're sewing clothes, you don't want that needle as part of your final product because it'll hurt you. So you cut off the needle. Much in the same way, the signal peptidase, very important enzyme, cleaves off the signal sequence. So it cleaves essentially right here. Cleaves right there, and the signal sequence goes away. And now you essentially have this final protein in white here, circled in red, in the rough ER where it's processed by different enzymes. Okay, things might be added like sugars. That's what a glycosyltransferase does. And then you can have different like redox reactions that are things like lysyl hydroxylase or prolyl hydroxylase. Just understand that there are enzymes in there that modify it. Okay, once the enzymatic modifications are complete, in the rough ER, the protein, the modified protein, is packaged into a vesicle, and the vesicle heads towards the Golgi apparatus. The Golgi apparatus accepts the vesicle, and then from there the protein is modified more by different enzymes, if need be. And then the Golgi apparatus packages it into another vesicle, and then the vesicle goes to different organelles or the plasma membrane where the protein will be inserted. Okay, I've mentioned cytochrome P450 in the past. Okay, cytochrome P450 enzymes do this as well. Okay, so hopefully this makes a little bit of sense. In the next video, we're going to go to a completely different topic. See you in the next video.